Hello, and welcome to this video on translation regulation. At the end of this video, you will be able to describe several ways that translation is regulated via processes that impact mRNA stability. The important part of this video is that just because an mRNA has been produced does not mean that that mRNA will be translated. So to talk about that, we're going to start with a little bit of review. Remember that mRNAs are processed before they can be exported, and that processing is coordinated by the C-terminal domain of RNA polymerase II, the series of amino acids that sit almost like a tail of that protein. Remember that that tail is made up of several repeating units, which include several serines, and that the phosphorylation of those serines provide binding sites for 5' capping enzymes, the spliceosome, and the polyadenylation factors. So one step at which we can regulate how much of an mRNA is prepared for translation is in the ways that this processing occurs and how efficiently they occur. mRNAs that are not properly capped and tailed will not have very long half-lives in the cytoplasm, as we'll see in a few slides. Additionally, those mRNAs have to be exported using the RAN-GTP cycle. Transcription occurs in the nucleus, while translation occurs in the cytoplasm. And so the rate of mRNA export and the efficiency of mRNA export can be used as a way of regulating how much mRNA is available for translation. Additionally, some mRNAs are not translated because they are regulated by the assembly of the ribosome. There are a few ways that the cell can use to prevent ribosome assembly. You'll see that in this picture, they depend on RNA secondary structures. These are often called stem loop structures because they include this self-complementary section, which make up the stem, and the part above that, which is not self-complementary and makes up the loop. So in the figure on the left, we see that the ribosome binding site is just downstream of a secondary stem loop structure. And that stem loop structure can actually provide a binding site for translational repressors. And so by having a protein that can bind to the translation start site, we essentially prevent any other ribosomes from binding. And only when that repressor is gone could translation of this protein begin. On the right, we see another stem loop, and in this case, the ribosome binding site is actually tucked into the stem loop itself. And what that means is that the ribosome will not be able to bind there right now. If we could somehow melt or otherwise take apart that secondary loop structure, the ribosomal binding site would become exposed, the ribosome could bind, and proteins could be made. This is particularly helpful if you have RNAs that create proteins that are temperature sensitive and therefore should only be produced when the temperature is increased. We also know from a previous video that translation can be regulated by small RNAs. Remember that small RNAs begin as double-stranded RNAs that are produced by transcription. They are processed and exported using RAN-GTP out to the cytoplasm, where they get picked up by a protein called RISC. RISC will choose one of those pieces of the double strand, and it will bring that piece of small RNA to the mRNAs. When the small RNA is fully complementary to the mRNA, that serves as a signal for a nuclease to degrade the mRNA itself and prevent future translation. When that small RNA is only partially complementary, and there's a section that is not complementary, rather than serving as a signal for nuclease-based degradation, that actually just serves as a way of preventing the ribosome from binding. You can imagine that if the ribosome was sitting here, translating and trying to translocate in this direction, it wouldn't be able to do it because there's already an RNA bound to the mRNA in that coding site. That might serve as a kind of temporary inhibition of translation. If that happens long enough, eventually those mRNAs will also be degraded by nucleases because they are not actively being translated. That process of mRNA degradation is highly regulated. mRNAs are degraded by nucleases that can recognize both the 5' and 3' ends of the mRNA, that is, the 5' phosphates and the 3' hydroxyl groups. So remember that we try to prevent this by adding a 7-methylguanosine cap in a 5' to 5' linkage. That 5' to 5' linkage actually cannot be recognized by nucleases and protects that segment of RNA. The poly A tail is a different kind of protection, but one that's really just a competition. These enzymes out here can recognize those poly A tails just fine, and they can chew them back. If they were to chew them back far enough, eventually it would start cutting into the mRNA itself and cleaving out coding regions of the mRNA. We can regulate the rate at which these mRNAs will be degraded by removing the 5' cap, which makes them susceptible to 5' degradation, or by shortening the poly A tail, which leads to 3' degradation that eats into the coding region. Again, the point is that just because an RNA is produced by transcription does not mean that it will be translated. There's a whole set of regulation that can occur between transcription and translation, meaning that the number of RNAs produced is not always correlated highly to the number of proteins that are produced.